this video, in this video we are going to understand the blood groups, the various type of blood groups. When we talk of human beings, there are two systems, that is the ABO group and the RH fat. There are two types we will be discussing about in this particular video. Now, the ABO blood grouping system was found by Karl Landsteiner. So, this system was found by Karl Landsteiner. Now, when we talk of the human blood, we talk of it in terms of blood group A, B, AB or O. Now, the difference in the blood is due to the presence or absence of certain protein molecules, which we call them as antigens or antibodies. Antigens actually are present on the surface of the red blood cells. So when we are talking of ABO grouping, we have to talk about the antigen and the antibodies. The antigen are present on the surface of RBC, and these antibodies are present in the plasma. Now, this results, there are different types of antibody and antigens. As a result, we have different types of blood groups. Now, the blood group which we acquire depends on what we have inherited from our parents. So now coming on to the blood groups, let us discuss about the types. We all know there are 4 types of blood groups when we classify them. The blood groups are A, B, AB and O. Now the blood groups actually are decided by the genes, that is the allelic pair. You know, each of the trait is governed by the allelic pair. So the genes over here which decide onto the blood group are also of allelic pair, and that is why we call them as we decide that depending upon that decides the type of blood group we are going to have. Now, if a person receives if the genetic composition is IA and IA or IA or IO, then a person will get blood group O. If a person has IB or IB and IO, then the B blood group. If a person has IA and IB, then the person gets a blood group of AB. And if it is I0, that is we write it normally as small I or small IO, then the person will be having a O blood group. So this decides, one from the father and one from the mother. Now if a person blood group is A, then the antigen which is present on RBC, this is the blood group, the antigen which is present on RBC obviously will be A type. Over here it will be B type. In this it will have antigen for A and antigen for B, because one is it is showing, actually this is codominance we call it, because neither of the allelic pair is dominant, both are equally dominant, so both will show the trait. And over here the last one, the RBC do not have any antigen, so it is nil over here. No antigen are present in this RBC. When we talk of antibodies, antibodies over here, A will have antibody against B. So it is anti-B. It will have against A. So it is anti-A. It will antibody against A. AB, since it is having both the antigens, it will have no antibodies in the plasma. And over here, since it is not having any antigen, it will have antibody for A as well as for B. Now when we look over here, you can make out from this table itself that during the transfusion, that means when the blood is given or taken, that is during the blood transfusions, why it is necessary to match with the blood groups? Because a person with blood group A has antibodies against B. So if a person with blood group A is given a blood of blood group B, then the body will consider it as a foreign body and will tend to destroy it, and the person will die. So therefore, complete matching of the blood group is very, very essential for all kinds of blood transfusions. Now over here, let us try and understand. A has antigen A and antibody B. B has antigen B and antibody A. AB has AB antigen, so nil antibodies because this plasma is exposed to both the types of antigen. And here, the plasma is not exposed to any type of antigen, so it has antibodies against A and B. So therefore, over here, individuals in blood group A can only be given to the recipients of A and B only. The A1 can give blood to A and O type only. The B will give it to B and who do not have the antibody against B, that is to person group B and AB. AB produces both A and B antigen, so therefore their blood can be transferred into people of blood group AB. So over here we can make out that people those who are having blood group O, they can donate the blood to any type of blood group, and hence the O group are called as universal donors. Person with AB group can accept blood from person with AB as well as other group, and therefore they are known as recipients, universal recipients. So AB group is universal recipient, and this group, that is AB, is universal recipient, and this is universal donor because it does not have any antigen. So it can give blood to anyone because it is not having antigen, so body will consider it as own and will accept, whereas AB can receive from all the types because it is having antigen both and no antibodies against any of the antigen. So therefore, O is a universal donor and AB is a universal recipient. Coming to the RH factor or RH blood grouping. It was again found out by Carl Steiner and Wiener and Levine. 3 scientists were there and they found out about the RH factor. The RH is actually the rhesus monkey. That is the rhesus, it was first of all reported in rhesus monkey. Therefore, the name RH factor. Now, nearly about 97% of India is having RH factor. this is an antigen. Actually, it is RH is an antigen, and it is also present on the RBC. A person who has that RH, the antigen on the body, on the RBC, will be known as RH positive, and the one who does not have the antigen is RH negative. So most of the Indians have RH factor present. Now, the problem is when when the transfusion is there, then the RH factor needs to be counted. Again, it has to be related to the, since it is a antigen, it has to be related. Otherwise, if there will be specific antibody formation and it will be rejected. So RH group also needs to be matched before the transfusion is made. Now, RH factor comes to special significance when we talk of pregnancy. Now, if a mother is RH negative, let's talk about a condition in which the mother is RH negative. RH negative mother, and the fetus is RH positive. This is a case. Now, in this case, immunological incompatibility happens. If this is the case, then it is a case of immunological incompatibility. Why we are saying immunological incompatibility? Because the RH factor is an antigen which is present on the RBC. Now, what happens is actually the RH antigen of the fetus is not it is not exposed to the RH negative blood of the mother in the case of first pregnancy, because the two bloods, that is the mother's blood and the fetus blood, is separated by placenta. So there is no interaction of these two type of blood forms, and as a result the first pregnancy normally remains unaffected. Now, during the delivery, what happens is during the delivery of the first child, quite a possibility that maternal blood get exposed to the RH positive blood of the fetus, and as a result the mother's blood will start forming the antibodies against the RH positive blood of the fetus. And later on, when the pregnancy happens for the second time, in case that mother's blood will interact with the RH negative blood of the fetus and destroys the fetal RBC. So what actually is happening, due to this incompatibility, the first pregnancy remains intact, that means it is not harming the fetus, but in the subsequent pregnancies, due to the exposure of the RH positive blood to the RH negative blood of the mother during the delivery of the child, the mother starts developing the antibodies against this. And when the second pregnancy happens, the mother is still RH negative, but it has developed antibodies against RH positive. This is the second case. This is the first case. Now this is the second case. So what happens if the RH positive is there, fetus, in the second pregnancy, this will be considered as a foreign body and the RBC of the fetus will be destroyed, will get destroyed due to the antibodies, and as a result the fetus will not be able to survive. Fetus does not survive. This is quite a complication, and it is known as, since there is a breaking of the RBC or the destruction of the RBC, it is quite fatal to the fetus, and this is known as erythroblastosis fetalis. Erythro is RBC, blastosis is breaking up or the degeneration of the fetus, that is fetalis. So this is arthroblastosis fetalis, and this condition is erythroblastosis fetalis, and it can be corrected or it can be treated by giving anti-RH antibodies to the mother soon after the birth of the first child, so that no antibodies are developed after this against RH positive fetus, and the pregnancy can then be continued. So the blood transfusion is an important phenomena where blood groups need to be concerned, ABO blood group and the RH factor.